we have roughly 290 intellectual disability genes described by carefully studying large monogenic families and using the best available methods at the time. But then from 2010 until today, this has exploded with over 1,000 different intellectual disability genes described. And actually, of those, at least 160 have treatable targets. And this is actually the case for around 500 rare disorders today, that there is some sort of available treatment. Some of them are super expensive, like gene therapies and bone marrow transplants, but some are cheap, like off-the-counter, non-expensive available drugs and nutritional supplements. And we can only offer these to patients if we know the molecular diagnosis. Because precision medicine really starts with precision diagnostics. Once we know the key puzzle to what the disease is caused by, we can then tell the parents or the individual why this happened. We can tell them if there is a recurrence risk and offer them proper surveillance and prenatal testing. We can tell them more and more about the prognosis, and more often, there are actually treatments or clinical trials available. So the problem is that even with the best possible genomic analysis today in the clinic, not somewhere in the vicinity of 35 to 40% get a diagnosis, and 60% remain undiagnosed. And around the globe today, many, many millions of individuals with rare disorders are still undiagnosed. So the European definition is that it's a condition occurring in less than 1 in 2,000 individuals. That means that there are 5,000 new cases in Sweden each year. There are 30 million people living in the EU with a rare disease. And these can be even those who get a clinical genome, which is a very small part of these 30 million, right? Only 40% get a diagnosis. But the others who don't even have access to that, they can have known conditions, but that where treatments are available, but they are not properly diagnosed. Roughly somewhere in the ballpark of 80% of the rare disorders are genetic. And really briefly talking about genetic variation, we have the single nucleotide variants, the single base pair changes. There are roughly 5 million of those in a human genome if we compare it to a reference. And 70, I can read my own slide, 3 of them on average are de novo 1 to 2 effect genes. And what we try to do clinically when we analyze these 5 million variants is that we filter out the low-hanging fruits here. We're looking for those truncating variants or those rare conserved missense variants that we think are detrimental to protein function. In addition, each genome, we have roughly 10,000 structural variants. So these can be balanced, like inversions and translocations, can be unbalanced deletions and duplications, going from 50 kb to megabases to full chromosomes, like trisomy 21 is sort of a whole chromosome duplication. And then we have these weird things, these special rearrangements like ring chromosomes and marker chromosomes and complex genomic rearrangements that also seem to be happening at different levels and are present in the background population variation. We have complex variations when we look at the genome in that way. But you won't find these variants unless you look for them. So if you do a clinical genome and you only look at the SNVs, you cannot say that it's normal because you haven't looked at this class of variants. The de novo rate here is a little bit lower. However, I think there's more to discover there, and with long read, we may be able to get a better estimate. So I talk about long read and short read, and this is just really briefly. Short reads we've been doing for over a decade now, where we do paired end sequencing of 150 base pairs on each side of roughly a 400 base pair insert. And now, since a couple of years back, we've been able to get long read data. What you see here is the Oxford Nanopore technology, but there's also another provider called PacBio that I'm not showing. And for both of them, in addition to getting really long reads of A, T, Cs, and Gs, we can get the 5-methyl C information, which is really interesting and something that we are now using both in our research and think could definitely be used also in diagnostics. So with that, I want to take a step back and talk to you briefly about the Swedish healthcare system, because I mean, all these different types of systems are different. In Sweden, we have a publicly funded and decentralized healthcare. So Sweden is regionalized. We have 21 regions, 290 municipalities, 7 university healthcare regions. So this is Region Stockholm, where I work, and I'm the director of the diagnostic lab there, where we service a population of roughly 2 million people. So the healthcare providers are either public, which is the majority, but some can be private, and then they're often contracted by the public. So it's very rare to have privately financed healthcare in Sweden. And genomics-based diagnostics is part of the specialized care, and it's usually based on referral from the primary care. So for instance, if I have clinic and I meet someone and I think that it's warranted with a clinical genome, I can order that, and it's funded by the public healthcare system in Region Stockholm. However, since we're regionalized, we actually have genetic diagnostic lab at the 7 university healthcare regions, and the system hasn't been harmonized. So we really don't have equal access to care in Sweden, and especially when it comes to personalized medicine and precision diagnostics and genetic diagnostics, this has been a mess. In 2017, we started the Genomic Medicine Sweden project to really harmonize and push towards equal access to genomics, and also push towards precision medicine in the entire nation. So this is a bottom-up initiative. So we did not like Genomics England, where they were like, okay, we're going to do this. This was more like, we want to do this. Can you please give us the money? And we've been trying to kind of pool our resources, and not, because if we are doing evaluations and implementation studies in several regions at the same time, it's obviously not, you know, a good cost benefit to the system. We worked a lot on national standards and guidelines and methods, and we built a common IT infrastructure in the Gothenburg region, which will be the clinical 1 million genomes node. I don't know if you know, but this is the European project that Claudia briefly mentioned, where we're planning to sequence, well, now we're beyond 1 million genomes in Europe, and really try to share data between nations in a federated and you know, way, but still be able to use that data for the best of the entire sort of European and probably global community. We have a number of clinical focus areas where rare diseases and hematology have been 2 of the most advanced when we started, and I think they are still sort of the leaders in the field when we compare to, for instance, complex disorders and other fields. And if we're talking about Genomic Medicine Sweden Rare Diseases, or GMSRD, I co-chair this initiative together with Hans and Lovisa that you can see here, but also Anna Vedal and Nogren have been really important collaborators in this initiative. And as I said, we started in 2017, and I think for the first 2 years, I really pushed to get a national ethical approval for a multi-center study where we can share data between sites, where we use the same informed consent in the entire nation for rare diseases research. And we really sort of also kind of brings the genetic community together in a common structure. And this was approved in 2019. Then we've done a number of pilot projects that haven't been enormous, and I'll talk to you a little bit more about the long read sequencing project later in this talk. But we started with just short read genomes, right? So we started in Stockholm in 2015, but at that time point in 2020, several regions in Sweden still didn't have clinical short read genomes. And just to kind of get everyone up to par and at that level where we think was the best for the patients at that time, this was our first pilot. We've been doing a big pilot on transcriptome analysis that we're pretty much ready to launch clinically now. And then long read sequencing was done last year. Currently, we have 2 projects. One is a really interesting digital consent project where we're using the ethical approval that we have from 2019. And we built a digital portal where, I don't know if you have bank ID here, but it's a digital sort of ID that everyone in Sweden pretty much have. So you can log into the portal with your digital ID, and give informed consent that way. And when we approached a cohort of sort of healthy adults or mentally normal adults who had done clinical genome sequencing, they got letters, they could log in and with their bank ID, 40% consented to sort of donate their genomes to research straight up. When we approached in a very small pilot, 20 parents of kids undergoing investigations for intellectual disability, 1 out of 20 was able to do this correctly because you need to go log in. Mom, give consent, dad, give consent, and then both need to log into the child and give their consent. So 4 different logins, and it becomes more complicated, and maybe it's easier to do under some sort of supervision. But still, this enables us to sort of, people with rare diseases, they want to share their data. They want us to do research on them. And this enables us to get consent from more people for that research that we are currently doing in a streamlined fashion. We also last week got funding, and I need to kind of calculate the budgets here, but like $1.5 million from the government to advance the diagnostics of undiagnosed rare disease children with intellectual disability or syndromes in this DMS Undiagnosed Network Sweden. But going back to the short-read genomes, what you're seeing here is a summary of what was done in 2021, roughly 4,000 genomes in total, and actually 3,000 of those were done in Stockholm, because that's where we started already in 2015. And I thought I'd take a few minutes and just tell you guys about the background and how we came to that point that we have a really well-functioning clinical genome, because it's a lot of work on different levels. I mean, we have gone through 4 different organizational structures with the Genomic Medicine Center Karolinska Rare Diseases, formally founded in 2017, but we've been tightly collaborating up until then. And this is a clinical academic partnership. It was the first time in Sweden we had anything like that, where patients from the healthcare system got access to the latest available sequencing technology through the universities. And then, we could report the results back. We have gone through a lot of sequencing instruments. We're currently using the NovaSeq X, as well as loads of bioinformatics softwares and forming pilots before a type of variant calling goes into clinical routine. So this is where we're at today. End of May 2024, we've reached 19,000 samples. We're going to reach the 20,000 samples in July, according to the prognosis. In 2 key papers in genome medicine, we describe both sort of the first 5 years of clinical genomes. There we had a 40% diagnostic yield. As we're seeing more and more samples with maybe less severe phenotypes, this tends to drop. But I think this is really where a lot of groups have been reporting. And the second paper is from my team, showing the work we did validating structural variant calling from clinical genomes back in 2019. This is the current setup. So basically patients, yes?
I wonder how many percent of patients are sequenced when they have a undiagnosed. The number seems to be a big number, right? But when it comes to everyone, like, you have a sense of how many percent of the total patients are being, or do anyone that needs sequenced get sequenced?
I would say no, definitely not. I mean, I think we have a huge task ahead of us, just educating healthcare personnel to identify, suspected rare diseases. Maybe AI will help, right? By kind of pinpointing when you have these 3 things together, think rare disease and sequence. So definitely not. Even in Stockholm, where we do 400 clinical genomes per month, loads of patients, even with intellectual disability, are not, sent for analysis. And we have a huge backlog because we have all the adults, right, who were kids at the time when we did a race, or maybe even before a race when we did karyotyping, who are still undiagnosed. So they are probably not preferred for testing unless something happens. So the simple answer is, even in a region like mine, where we have a very well-functioning clinical genome, everyone who would warrant a clinical genome does not get referred for testing, even if it's government funded. What I try to draw out here is a little bit how we work. So patients are obviously seen on the healthcare side, where we not only sample them, but we also map out the family. We do the different types of clinical tests that are necessary in a normal diagnostic procedure. Then the clinical sequencing is done, and that is very streamlined now and quality assured. And we call all these different variant types, and finally we get a ranked kind of called variant file that's visualized in an online variant interpretation system called Scout. So there I can log in from the clinical lab, my people who work there logs in, but we also have clinical experts, physicians who can log into the system after training and help with variant interpretation. Because I think it's important to remember that you cannot make a diagnosis by genetics alone today. We don't know enough about penetrance and sensitivity of gene variants. So we always make a diagnosis together with a clinical presentation in the actual individual that we have tested.
Respect to that, what about cascading testing? How does that work in this ecosystem? For example, if somebody had a sibling disorder or metabolic disease.
So that would be somewhere where clinical genetics obviously would try to be helpful. We cannot contact relatives of people with disease. So the contact has to be through the family, and then they can contact us or get a referral to our clinic and get tested.
Would it be genome or would it be targeted?
That would likely be targeted. I guess it's a different story. We still do. I think a lot of people think that, in genetics today, it's only genomics, right? But we do tons of Sanger still in the clinical lab. We do so much MLPA. We've had targeted small gene panels that we're slowly retiring and moving to the genome. And I think the cost of the genome versus the personnel cost of these targeted tests. And as the short read genome is reducing in cost, I'm guessing that will likely take over more and more on what we're doing in the sort of diagnostic lab eventually. At the same time, I think when we get that prenatal test, it's really nice to have the primers in the lab already and the assays set up, compared to having to rely on a sequencing center and those instruments. It's very nice to have it quick and ready. So it's a balance. What's reported out clinically are the ACMG 4 and 5s and few VUSs where we really think that there's a test that can kind of push that one way or another. These are a few papers from my group in the past 2 years where we sort of evaluated the genome sequencing of specific cohorts, such as neuromuscular disorders or prenatally detected fetuses with malformations or intellectual disability, where I work a lot. We also looked at the cost effectiveness of genetic testing in intellectual disability as a first-line test. And surprise, surprise, it actually saves money for the healthcare system because you get a faster diagnosis. The diagnostic yield now, when we're at almost 19,000 patients analyzed, is roughly 22%. And it ranges a lot between disease groups. The highest yield is in the skeletal dysplasia panel, and these kids are very, very carefully selected and often X-rayed in multiple different ways before they do the genome testing. And there we have a 44% yield, while in the connective tissue disorders group, the genetic testing is done more to kind of both to exclude genetic disease and find them. And there we have a 9% yield. Now, I start talking about structural variants, and I like structural variants. And if we look at this slide, I went over the data from roughly 1,000 individuals with neurological disease. So these can be neuromuscular disease or intellectual disability and developmental delay. And in those individuals, we found a pathogenic variant in 26%, so that's 288 individuals. And you can see here that 30% of those pathogenic variants were non-SNVs. So if we had only done SNV calling, we wouldn't have detected them. However, even more disturbing, so these are, again, severely affected individuals, and then 70%, they are still undiagnosed after a short-read genome with, multiple variant type calling. That said, maybe I didn't say that in the former slide. So clinically, we use in silico panels where we look at clinically relevant genes in that specific phenotypic presentation. So if there's a new gene that hasn't been described, we wouldn't pick it up. Then we would have to open up and look at the whole data in a research context. However, one thing that people have been, talking about a lot lately, and obviously that we're also interested in, and that I talked about in my introduction, is long-read sequencing. And we've made within the Genomic Medicine Sweden consortia, we've made somewhat of a very broad 5-year plan for how we see that long-read genome sequencing can enter the rare disease diagnostic space, starting right now with verification of repeat expansions and structural variants. Then we want to quickly move to kind of screening and being able to digitally karyotype and find these chromosomal rearrangements with the long-read sequencing, but also analyze, obviously, pseudogenes and repeat-rich regions. And then moving to screening for the imprinting disorders, as well as conditions with high genetic diversity that I just showed, like neuromuscular disorders and intellectual disability. And then replacing, I think, more and more short-read genome tests in a screening basis, because I think if this test is even slightly better than short-read, it makes more sense to do long-read than to do short-read and then computationally try to reassemble a genome from that. I mean, we can discuss that also later over lunch, but I think that the field will move towards long-read now that they are available. And as the throughput increases, this is what we will want to do in rare disease diagnostics. So we decided in Genomic Medicine Sweden to try to validate long-read genome sequencing as a follow-up analysis of chromosomal rearrangements detected by karyotyping, array, or short-read genomes. And here's just a plot of the first 14 samples. These 3 here, this is the read length, and these 3 came from my lab where we had done a high molecular DNA prep. So you can see that you do get like tighter, curves and longer reads there. But we actually got really, really nice data also from the regular clinical DNA. And I think it's important to remember that when you're launching a clinical test, you cannot be about having the best possible QC values. You have to be about the result and the diagnosis and the patient. And the sample we get from that really sick kid is what we need to use, regardless of the DNA integrity value. So very briefly, in this small study, we resolved 4 translocations, 2 inversions, 6 complex genomic rearrangements, and 1 deletion. And here we might have a bit of an enrichment of complex rearrangements because we basically went out to the centers in Sweden and said, give us, your toughest cases and see what we can do with long-read. So obviously quite a few of them were hard to had a lot of complexity going on. We had 2 events that we couldn't resolve fully. One mosaic ring and marker chromosome 21, and you can see the phase analysis here. And obviously, when it's mosaic, you're going to have a lower read depth for the specific event. And the 21 is one of these chromosomes where the acrocentric arms have a lot of repetitive regions, and that is quite hard to resolve still for us. So we're still struggling with that and actually getting a new sample to be able to do a high molecular prep and maybe a de novo assembly. We also had a suspected disomy 15, which you can see here with like a tetra and trisomic regions close to the centromere. And this is from the short read data. Here we could also see the same kind of read depth differences in the long read data. However, long read data is not as good as short read for that type of an analysis yet, but we couldn't capture the actual breakpoints. So just to give you some examples of what the data look like, here you have a karyotype with actually two events. There's a translocation between chromosome 1 and 10, and then there's an inversion on chromosome 2. And this is a circus plot where you can see the translocation between 1 and 10 and the inversion very clearly. These are screenshots from IGV, and you can see these really nice, long, soft clip reads really indicating the presence of the junction. These 3 genes that were disrupted have not been linked to human disease, and this was found in a mother and a daughter with infertility problems, which is common when you have these types of structural variants that can, through errors in during gametogenesis, cause unbalanced gametes to arise, right? So you're not expecting them to have any monogenic disease genes since they didn't have that kind of a phenotype. The next case is a syndromic child with a craniofacial condition. And here we saw already in the array that there was something weird going on chromosome 2. The karyotype, however, was normal in this case. And this chromosome 2, there was a dupe of the B segment, triplication of the C segment, quadruplication of D, triplication, duplication, deletion. Wow, right? Then we look really carefully at the data, and we see that there's a separate CGR on chromosome 2q with a deletion, inversion, deletion. So in total, 4 megabases of sequence are deleted, and likely the phenotype is caused by the combined effect of all of those genes in the deletion, maybe also some of the balance junctions. But this is a little bit like the more you see, the more you look, or Finding Waldo or something like that, right? That you look at this data, I'm like, what is going on here? Oh, it's a second complex rearrangement in this patient. I also wanted to highlight this case. It's the karyotype here was done on bone marrow chromosomes, and you can see bone marrow chromosomes are always very ugly and hard to interpret because the patient had thrombocytopenia. But there's actually a paint done here, so you can see in red 3 chromosomes X, and one of those 3 is a derivative X with chromosome 9 material on it. And here you have the other 9. There's a little bit of X there, but you can't see it with the paint. And here we couldn't capture the junction in GRCH38. We had to use T2T to get to the right position in this very repetitive region. And this is also something to think about, right? I said, if you do a genome and you only look at SNVs and you don't find anything, you can't say that it, has to be environmental. And the same goes for if you do a genome and you only analyze it with GRCH38 and you don't find anything, you may still have more experiments or analysis to do, right? So this study was done together with the Uppsala team, and we used the PacBio Revio. It was one of the first instruments that was delivered to Europe. That was installed in Uppsala in May last year, and Lars Folk here and Adam were really, really important, as well as my senior bioinformatician, Jasper, who did a lot of the analysis work. So with that, I wanted to kind of follow up on this reference story and tell you about some work that me and Claudia are doing right now on inversions. This is an inversion 6 with 1 breakpoint on the P arm and 1 breakpoint on the Q arm that we've been struggling for quite some time. And we actually, in addition to short read genome sequencing, we also did a long read, and we couldn't find the breakpoints. And we were like, okay, do we need to do HiC? How can we capture this? What's going on? And then one of my PhD students, Christine, remapped the data from HD19 to GRCH38. And now we see the junctions in both the short read and long read data. So this is kind of an expensive way of finding a rearrangement going all the way through long read here. The reason is that there's actually a gap in HD19. So there's 227 kb that are present in GRCH38, but are not there in 19. Now, if we compare this to these 127 kb are present in T2T and also in chimpanzee and bonobo. So any type of primate reference genome out there would have been better than HG19 to analyze this short read data and find this inversion. This inversions are interesting, right? Because you have two junctions in cis, and you need to capture both to capture the event. And it's easy to say that, oh, well, you probably found, this one, so why bother with this one at all, right? But I think we have so many variants, we have 10,000 variants. So we're always going to have, signs of disruptions at multiple places that we don't know what to make of. So we really need to find both to know that we have an inversion there. Claudia and I did a study a few years ago looking at cytogenetically visible inversions, and we could find 13 of the 18 with short read and HG19. And we hypothesized that these 5, that we couldn't find, maybe the junctions were present in large repeats that we couldn't span, and maybe there was some non-allelic homologous recombination, etc. And we decided to go back and look at these with multiple reference genomes, and also when we had DNA available to do long read sequencing and de novo assembly. We got an additional case from collaborators at Baylor, and we also, just this morning, got an additional case from the Karolinska that I don't have in these slides. So right now we have 7. So this case from the US is actually an inversion 9 that happened de novo in a child with a clinical picture, including a syndromatic presentation, basically. And when we map this data to T2T, we find that one of the junctions disrupts the EHMT1 gene. And the diagnosis here is actually cleft stress syndrome 1. So, but why couldn't we find it? So they did short read and tried to map it and couldn't find the inversion breakpoints. And the reason again is that the region is not present in this case in GRC38, but only in T2T. And there's 29 megabases missing from the reference in this breakpoint close to the centromere that consists of 99% satellite repeats. So here we have a monogenic disease being caused by a de novo translocation with one of the breakpoints in a junction that required T2T to be resolved. So this will be presented at European Society of Human Genetics as a poster if you want to come and talk to Christine about her data. We had a new inversion, like I said, this morning that we couldn't resolve in HG19. And when we looked at it, it's present in T2T and in chimpanzee and in bonobo. So this was a case of infertility. So this seems to be important for inversions for sure, maybe also for other types of structural variants. And with the balanced rearrangements, translocations, and inversions, we need to find both junctions in order to call them properly. So I tried to show you that even if we're struggling a bit with the reference and colors and so, we are able to use long read genome sequencing to verify chromosomal rearrangements in many cases. However, if we want to introduce this clinically, we also want to use it as a screening tool. Also, when you're introducing a method clinically, there are a lot of quirks you need to kind of solve along the way. You need to have a streamlined system that is sort of stress tested. So last summer, together, we discussed with Oxford and Nanopore, and we decided to do a parallel study of fresh cases, basically. So 100 cases coming to the Karolinska that are referred for short read genome sequencing, we're going to do long read genome sequencing in parallel. And we're using the Oxford Nanopore PromethION for this. We're using regular DNA, so we're not doing a high molecular extraction. I think it is obviously better if you can get longer reads, but at the same time, if you can get a couple of thousands of nucleotides, it's still a lot better than 150, right? We built a pipeline called PorePipe, and it calls SNVs, SVs, STRs, and also methylation. And the data is visualized in this browser-based system for variant interpretation called Scout, where we also interpret our short read data. We chose to include patients with intellectual disability, developmental delay, but also with those with neurological disorders. If we're looking at the first 50 cases here, the main phenotype, most of them had neurodevelopmental delay or global developmental delay. Some have a more syndromic presentation, and then we had a fraction with more muscle weakness and walking abnormalities. We have quite a few SVs if we're looking at the short read data results of these first 50. However, some of these VUS those are mostly in the SNV indels. So I think this fraction is going to grow a bit. So we're going to end up with more or less the same numbers that we are used to seeing with somewhere in the ballpark of 15% SVs or so. Looking at the QC, and we tried a couple of different ways of fragmenting the DNA along the way. So in turquoise, you have fragments with the megaraptor system, where you use controlled hydrodynamic shearing. I have no idea what this is, but apparently, you don't damage the DNA as much by pressing it through a microfluidic chip than with, more mechanical shearing versions. And we can see that the median read length goes up, but the median coverage almost goes down. And this has been well described. Like if you have shorter fragments, you tend to have higher read depth. So our median coverage is really nice for all of these samples since the start. The median length is lower than what you would expect in a scientific study, right? But again, these are clinical samples. This is the DNA we have, and this is the reality we live in. Looking at SNV, so far we found 100% of the SNV indels that were pathogenic, and they were highly ranked and easy to find. On the left here, you can see an HEVAP2 variant, which is an intellectual disability gene, and it's nicely phased. I don't know if you can actually see it, but it is nicely phased. You're going to have to trust me on that. While on the right side, we had slightly shorter reads and wasn't able to phase the variant, but we could still capture this variant very nicely. Looking at structural variants that I love, we have 9 structural variants to date, and here you can see A22Q11 duplication by redapt and B allele plot by CNV Piter and the HiFi CNV. You also get the coverage track here. We are not able to bridge across the 22Q11 low copy repeats that promote the formation of this rearrangement. So in that case, we don't have long enough reads, but we can capture even this type of an event. And I think the health economy in these types of tests always comes when you can replace something, so you don't have to do multiple testing in specific individuals. Here is a really interesting chromosome 18 rearrangement. Starting with looking at the read-depth plot, you can see that there's there seems to be a loss of chromosome 18p. And when we karyotype, we see this very nicely, that we've lost chromosome 18p. It's also visible in the small blurs here that it might be harder to see. And we see these in 31 cells. And then we have 8 cells where we have a ring 18. And if we look here, it looks like there's some sort of mosaic thing maybe going on 18q. And we can nicely capture the split reads between 18q and p and closing the ring directly with a long read sequence. I think that's kind of cool, right? It's really digital karyotyping here. Next example, a complex chromosome 9 rearrangement, where we have a deletion and then a duplication here in the short read data. We look at the long read data. Again, we see the deletion and the duplication, and we can also nicely see the junctions with these nice long split reads. And here you have the assembly where we have a deletion, normal, inverted duplication pattern. We can actually see in the short read read-up plot that there is evidence of this normal segment, even if we didn't see it here. And we can, if we zoom in, actually read into the telomeric sequencing with the long reads. And this is exactly the model that's been proposed for inverted duplication terminal deletions by the Rudd group like 10 years ago. So I think this is really interesting how we, with the new sequencing technologies, can just directly capture these types of rearrangements that have been proposed previously in the literature. So we had a total of 9 rearrangements. Of these, 4 are complex. And these are fresh cases. And I really think that complex rearrangements, me and Claudia, we had a review article on that last year, right? Are more common than you think. And really, when you zoom in and you map the junctions and you try to resolve the rearrangements, we're finding that complexities are very, very common. So I'm talking too slowly, obviously. I wanted to have time to also talk a little bit about methylation, because this is really cool, right? With long read, we don't just get the ACTGs, but we also get the 5 methylations. And one example here, remember the X9 translocation in the GMS RD project? Well, here you can see translocation X9, here you can see control, and if, let's see, if it's blue, it's non-methylated and active. If it's red, it's inactive. And you can see in this phased sample that we tend to have this allele more active. You can also see it here by another program called Methyl Artist. And actually this allele with lower methylation is the derivative X, which is supposed to be active in an X autosome translocation. So this is a very fancy way of testing for X inactivation, but still cool that it works. Looking at the Karolinska Oxford Nanopore study, for the first 40 cases, we looked at the phase data and looked for allele-specific methylations driven by Jesper Eisfeld in my group. So here we looked at the promoters of genes. And you can just see a global chart here where we have in black, I think it's almost impossible to see anything, but here you have the Prader-Willi, Angelman locus. You have these very clear imprinted loci, right, that are almost showing allele-specific methylation in the entire cohort. And then, but we have genes with allele-specific methylation in all the autosomes. These are the first 40 patients and the top 200 genes, and we have the imprinted genes that then we have commonly imprinted genes and genes that are very rarely showing this allele-specific methylation, less than 5 individuals in our cohort. If we overlap this with the gene imprint database, we have a lot of candidate potentially novel imprinting genes by this analysis. The reason we're not seeing all of these is that we only looked at blood, right? And we also looked at the promoters of protein coding genes. So if the regulation is further away, we're going to miss it with our current approach. These 26 are all very well-known imprinted genes like SNRPN, etc. And here are 2 examples. HRT-R5A was showing allele-specific methylation in our entire cohort. It's not part of the gene imprinting database. While EVPL, that is part of this, was showing ASM in less than 5 individuals in our cohort.
I want to make sure I understand this. So for the imprinted genes, are they always associated with the X chromosome or associated with the X chromosome?
Not at all. These are autosomes.
These are
so
all
imprinting are if, an allele is turned on or off, depending on whether it comes from mom or dad. And we are actually not looking at X and Y in this. We're only looking at the autosomes. So the X translocation was sort of a different story, just showing that we can see whether or not it's inactivated by long read.
And so the syndrome results because it's a combination of imprinting together with, for example, SMB.
So that could be, that's one way of using this data, right? This would be a way of solving patients. Maybe we can have allele-specific methylation on one allele, and the expressed allele has a nonsense variant, and it's a recessive disorder. This could, help diagnose that patient that we would miss with our current pipelines.
Because that would look heterozygous overwhelmed.
Yes, exactly.
So then you would miss that without the methylation data.
Yes.
Okay.
So I wanted to take a few minutes. Claudia said in her introduction a little bit about what we do in the rare diseases group that I co-lead together with my very close colleague, Ann Nordgren. And we really try to take a holistic view of rare disorders. We have psychologists studying the disorders in both kids and adults. We're trying to push diagnostics like I showed, but also perform functional studies and on new and known disorders to understand more about the the mechanism of disease. One of the disorders that we have studied quite a lot is Williams syndrome. This is a recurrent deletion on chromosome 7q, where they have cardiovascular disease, developmental delay, intellectual disability, and it's really not a clear genotype-phenotype link yet. Here you can see a plot of all the genes that are deleted in the common region, and the red ones are those that have been previously suggested to maybe be part of the phenotype. The psychologist team in our group have done a lot of studies, even on older Williams syndrome cases, for instance, showing that if you have a lower IQ, you have a higher tendency to have anxiety. And we were able to get skin biopsies from 4 of these individuals and induced iPS cells and neurons. So one of my PhD students is currently studying the transcriptomic landscape of early neurodevelopment in Williams syndrome, where she took the 4 Williams cases, NESCs, neurons at 4 weeks and neurons at 8 weeks, together with 4 controls, and we're looking for differentially expressed genes. Here you can see a PCA plot showing that the nest cells cluster on one side and the 4-week neurons and 8-week neurons cluster here. And here you can see the neuromarker genes, where you again have nest cells, mix of cases and controls. Here are the nest cell markers. And then you have the neurons clustering together, again, mix of cases and controls, and here are the neuronal markers. If we zoom in on the Williams syndrome region, we can see at all 3 time points, lower expression of the Williams syndrome genes. And if we look at the differentially expressed genes, we have a lot of genes at all time points, and we also actually did blood that differ between them. When we do a GO pathway analysis, you can see that there are loads of pathways coming up as significant, especially in the 8-week neurons, and a lot of them have to do with CNS and neurodevelopmental pathways. Zooming in on 3 genes that have been, that are in the Williams region and have been suggested to maybe be part of the phenotype, we can see that we are seeing disturbances in these pathways, both the LIMC1 pathway, FZD9, and STX1A. This is also somewhat expected, right? Because it is Williams syndrome and they are deleted. However, the conclusion so far is that we see many different pathways and gene networks that seem to be sort of working together to cause the specific cognitive problem called Williams syndrome. Finally, I just wanted to take a minute to tell you about an event we organized last summer. It was my colleague Anne, together with the Karolinska UDP, the Willem Foundation, and Phenotypes, who decided to have an undiagnosed disease hackathon. And we had 100 experts coming in from all over the world and working together to try to solve these 10 patients from 8 different countries. And we got samples, we got a very detailed phenotype information. And before the hackathon even started, we did short read sequencing, we did long read sequencing, we did RNA sequencing, and methylation rates. And then we all sat together at the Karolinska for 72 hours and really analyzed this data. So we were divided into rooms with different experts in different rooms and really trying to see what's going on. Which is actually one of the most fun meetings I've ever been to, because often when you're at a meeting, it's sort of scientific entertainment, and I want to show, how cool my lab is, and Amy shows how cool lab her is, and so. But here we were really, it wasn't about that. It was about solving these cases and using our expertise. And we had Vincente there who wrote the drop pipeline, and we had been thinking about installing that, but we actually got it installed, at our house to analyze RNA-seq data. But it was just so, I don't know, like it was so much fun and such a nice meeting to be at. All in all, 4 patients were solved. And this was really, I have to say, a teamwork. I think also for the scientists who took part in this, the patients from Sweden, they came there. And they were then also both clinically assessed by international experts, but got to meet the scientists. And I think this was, for everyone, such a wonderful kind of experience. And with this, I just want to wrap up. I tried to show you a little bit how we do clinical genome sequencing We use a very defined, clinically relevant panels to try to offer precision medicine to those patients who can get diagnosed. But then we move on and do different types of studies to diagnose more patients and increase the diagnostic yield. And I often finish up by this quote from the mothers of one of our patients who we did an interview on Swedish radio last year. And she said that ask your doctor for a gene test. And if you have already had one of those, you should ask for a new test every 5 years because new discoveries are made every day. What you could not diagnose 5 years ago, you can do today. And I think this is extremely important that the expiration date on the testing we're doing is like not even 24 hours. I obviously did very little of this work, and all of it was done by my super talented team members, all of my wonderful collaborators, and I'm happy to take questions.
I'm wondering, I guess I also know US public health system, but for the Sweden, it seems to be a little different. Who's paying for the sequencing? Are the insurance company involved at all? And
No.
what's the future?
Yeah, so we have a publicly funded healthcare in Sweden. So this is paid for by the taxes that we basically pay, all of us. Yeah, it is kind of nice, actually.
Tax rate.
I mean, the tax rate here is pretty high as well, right? So
Because I have the impression, like at the beginning when I asked, like, I have a feeling that in Northern Europe, a lot of things are like, if people need it, they get it. I have that feeling.
I think there's a lot of improvements that can be made in that way. And like I said before, as we're moving into precision medicine, precision diagnostics, we need to educate our colleagues in the healthcare system to really be aware of this and to recognize the symptoms when they should refer patients. I mean, we obviously, clinical genetics is a small specialty. We won't be able to see everyone with a rare disease. So we have to have experts out in the different areas who recognize and when to send referrals. And we need to work together to sort of capitalize on each other's expertise.
Anna, I have a question. So you are doing a lot of new work and cohort, like already connecting the research and the clinic at the same time. So this is really, really nice. So where do you think is going after methylation? Where are you guys going?
I mean, I still think that one of the things, when it comes to diagnostic yield in rare disease, one of the things that we are struggling with is really the truly diagenic diseases. And in order to capture that, we need such big numbers of patients where we can do joint analysis to find these connections. There was also a really fun paper just a few weeks ago showing that a long non-coding RNA actually causes like 0.5% of intellectual disability. So in 2024, we can have such a discovery. It was by joint forces by Genomics England and Broad. And I would think that's where we need to go. We need bigger numbers and analyze our cohorts in that way. But obviously we need, I don't know if a long non-coding RNA is really, the non-coding genome, but I guess it is, right? And that's the space we need to move into and understand more about what outside of the genes is causing disease.
A question about the imprinting and looking at the methylation. Are you able to, so do you need the specific tissue that is affected for that? And would it be possible to do that in like the neurological development problems, or is the methylation patterns, the imprinting that you're seeing, sort of a whole person-wide methylation pattern of the imprinting?
As far as I understand, it's highly variable between tissues. So as always, we're looking for the low-hanging fruit. I mean, I love DNA. DNA is stable. It's pretty much the same in all tissues, and RNA is a mess, right? methylation is probably going to be similar, but I mean, we are seeing that if we look at blood or fibroblasts, I mean, it's not too bad. We can find a lot of things there. Then obviously, if you really want to be disease specific for intellectual disability, it would be much better to have neurons to induce IPS neurons in every patient with intellectual disability. I don't think it's a good use of healthcare money, but now there are more rapid inducement protocols that might be used for certain undiagnosed cases, even clinically. We just need to make sure that these truly reflect the methylation status of the tissue at hand, because I think a lot of reprogramming is happening. The methylation is bound to be affected somehow. So we're looking at the right thing, right?
I have a question in the chat. Can you see it or would you like me to read it out?
Oh, God.
Read it out.
Yeah, please do.
Did you have any cases where you had to combine with other technologies, i.e. optical genome mapping? Also, do you have any experience with signatures from long-range sequencing data? Thank you for your excellent presentation.
So we started using optical genome mapping for FSHD, which is like a weird repeat disorder, I think like 2 years ago. And now we are currently looking into optical genome mapping for myelomas and leukemias, but also obviously as a follow-up analysis for various structural variants in the rare disease space. I don't know if that's an answer to that question, but I think it fits very nicely in with the genomics technologies as a sort of validation by another type of technology, and where you can get more of a chromosomal kind of point of view. And the second part of the question was EpiSign, and I think this is super interesting. I haven't used EpiSign myself, and I think to do that in your own lab, you would have to have a good database of positives to recreate these kind of signatures. But it could be that we may be able to do it with less samples. But as far as I understand, you need to have like at least 10 people with that particular disease to make a signature before you can start using it. But I think definitely that could be interesting for some of the unsolved, and also to characterize if genes have episignatures that we just don't know about, right? So to systematically try to collect samples from individuals with various rare disorders and check these things is definitely something we should be doing as a community.
And then a second question, which is how difficult is it to filter your data for structural variants as we currently lack relevant databases?
Yeah, we have 19,000 samples in our database, which makes it easier. I think this is one of the problems today, that if you have, if you're a little bit older in the game, you're going to have an advantage to someone who is starting out. We also actually, the Swedish Genome Program, which was 1,000 individuals sequenced on the X instruments quite early, we performed structural variant calling on them, and that is publicly available, and it's quite an unfiltered structural variant resource for short read that we used in the early days as a normal variant database. Now, together with Adam Ammar, who's here, and we are in the Genome of Europe project in Sweden. Now I'm planning to do a read sequencing of 500 individuals, where definitely structural variant calling will be super important. And I think having the unfiltered structural variant files can be very helpful for labs who are starting to use a new technology to be able to filter out the rare calls, for sure. But the in-house database, I guess, is the short answer, that is extremely helpful in those cases.
So with the long-read sequencing data, are you able to use that to actually guide the short-read sequencing data? For example, I guess to either reduce the cost or sometimes look back at the older short-read sequencing data, right? So now you learn so much about the long reads, and then with that learned can be, sort of glean some information so that it guides us how to use the short data.
I wrote a grant on that 2 years ago. It was unfortunately not approved, but yes, obviously this is one way of doing it, that we learn from the long reads and we improve the short read. And I think to some extent, the technocratic sort of directive, the short reads are here to stay for quite some time for certain questions, I think. And maybe long reads will be used in certain areas, right? But we can optimize and improve the short read analysis for sure by learning more about our genome and how to look at it, and also knowing where short reads are good and where we might need to complement with other technologies. And I know people have discussed a lot about like what is the optimal kind of investigation if we have a child with developmental delay. Do we want to do a shallow long read genome together with the short read to get the depth? Now, as prices are going down, maybe it will instead be a deep long read genome. It's a little bit hard to say, but I'm sorry. But I think, yes, definitely, it's an interplay.
So thank you, everyone. Thank you, Anna, once more.
Thank you.